Good afternoon, everyone. I welcome you back to my YouTube channel. Presenting before you this wonderful tutorial is the mentor Hamper Kliman. And of course, if you are new to this YouTube channel, consider subscribing, hitting the like button, and don't forget to share to others. We come with another session where we will have to revise Bio 1-1 Molecules and Cells of 2024 Test 1 paper. And of course, we shall be doing our section A. Then in the other session, of course, you see it on this same YouTube channel. You will need to go through it. Okay, so we can start. Okay, so we can start. The first question is saying, which group contains both a carbonyl and a hydroxyl group? Which group contains both a carbonyl and a hydroxyl group? So when you look at the first one is saying alcohols of course don't, aldehydes don't, because an aldehyde contains something like this, okay, and this particular carbonyl group should be at the far end of a carbon atom. If it is in the middle, then it becomes a ketone. But then for a carboxylic acid, it makes sense because the structure of a carboxylic acid is like this, okay. This is how it is. So this implies that you're going to have a carbon. This carbon can be attached to a hydrogen there, a hydrogen there, a hydrogen there, and at the end you're going to see it attached to an oxygen atom, to another carbon atom, like that, and it's going to have a carboxylic acid functional group. So you discover that you're going to have this part which is a carbonyl and this part which is a hydroxyl group. Okay, so this one, our answer for this one is simply A. And of course the person got it right. I do not have a paper where there are no answers. Of course, some are wrong, but we shall be able to correct those that are wrong. The second question is saying, what type of intermolecular force is primarily responsible for the high boiling point of water? So when you look at the structure of water, water is just H2O, and of course this is just hydrogen, and then you are going to have oxygen, then hydrogen. These are hydrogen bonds. So the hydrogen bonds are the ones that are responsible for the high boiling point of water. Take note. Okay. Question 3. Question 3 is saying, in which scenario would you expect dipole-dipole interactions? Dipole-dipole interactions. So dipole-dipole interaction is particularly found in polar molecules. Okay, you only find it in polar molecules. Okay, for London dispersion forces, of course, those ones you can find them both in polar and non-polar. But whenever you're talking about a dipole-dipole interaction, consider polar molecules. So it can only be found between polar molecules. Okay, that is one important concept to understand. We go to question 4. In which of the following scenarios would you expect dipole-dipole forces to be the strongest? So dipole-dipole forces would only be strong on the dependence of the electronegativity of molecules. Okay, electronegativity of molecules. For example, when you look at the interaction between, let's say in H2O, you discover that the oxygen has got more electronegativity compared to hydrogen, because possibly hydrogen only has one electron in its outermost shell. Okay, in its outermost shell. But for oxygen, you discover that it may have of course, when it loses one, it will have 8. When oxygen, because it's got a partial negative charge, this guy's got a partial positive charge. So when this guy donates, this guy gains, you discover that when this guy stabilizes, it's going to have 8 electrons in its outermost shell, eventually increasing in its electronegativity. So when atoms have got high electronegativity, just know that they are going to have very strong dipole forces. It's very important. So it's between two molecules with large electronegativity differences. We go to question 5. So question 5 is saying, which of the following class is characterized by a carbonyl group bonded to at least one hydrogen atom? Which of the following class is characterized by a carbonyl group? So this is a carbonyl group bonded to at least one hydrogen atom. Okay, one hydrogen atom. So when you look at this, so when you look at this, ketones are bonded in between. So when you have, let's say, structure like this, we're going to have a structure like this. Okay, possibly you might have something at the end. You may have something like this maybe at the end. Okay, let's just say hydrogen here, let's say hydrogen here. I don't know which structure, but for a ketone body, you imply that you are going to have a carbonyl group in between the carbon atom. But for an aldehyde, okay, so this guy here will only contain one hydrogen. By the way, because it's a double bond, you won't even have a hydrogen atom here. Okay, but the question is saying is bonded to at least one hydrogen, so it should be at least more than one hydrogen atom. So more than one hydrogen atom, you are going to have something like this. It should be at the far end. When it is at the far end, that is when it can only be bonded to more than one hydrogen, because this is a bond. You've got this particular bond, you've got this particular bond there, all of those. But when it comes to a ketone, it's only found in between. And you only expect it to be attached to this unless it is broken down. That is when you would be able to have another bond attached somewhere there. So the best answer is an aldehyde. We go to question 6. Question 6, they're saying what functional group is formed when an alcohol reacts with a carboxylic acid? So when an alcohol reacts with a carboxylic acid, it forms an ester. Okay, esters are used for in perfumes because they've got a very good scent. Okay, so you form an ester. Okay, you form an ester. We go to question 7. Question 7 is saying which molecule has polar covalent bond. So polar covalent bond, they are implying one which has got a positive partial charge, negative partial charge. This guy here only has a negative partial charge. This guy may have a positive partial charge. This guy is non-polar. But when you look at hydrochloric acid, it's got hydrogen which has got a positive partial charge, and at the same time it's got a chloride which has got a negative partial charge. So these guys are polar. Henceforth, hydrochloric acid has got a polar covalent bond. We go to question 8. What type of bond holds the atoms together in a compound like water? So like I mentioned at first, here we are simply discussing bonding. Of course, we've got ionic bond, covalent bond, metallic, hydrogen bond. What type of bond holds the atoms together in a compound like water? Okay, so of course don't be misled. In water, of course the type of bonding there is a covalent bond. Okay, it's a covalent bond. That is one thing that you must be able to understand. Okay, so it's supposed to be a covalent bond. Please don't go with hydrogen bond because you are going to miss it. Okay, this oxygen, remember, covalent bonding is all about sharing of electrons. So of course oxygen is unstable, hydrogen is also unstable, so they both will need to share, and eventually they need to become stabilized. Okay, they need to achieve an octet rule. That is a very important concept to understand. We go to question 9. So question 9, they're saying, which of the following substances is considered an element? This is a compound, this is an element, this is a compound. So the best answer is simply C. We go to question 10. Question 10 is saying, which property of ionic compounds makes them suitable for use in biological buffers? Okay, which property of ionic compounds makes them suitable for use in biological buffers? So a buffer is simply anything that resists change in pH. Resist change in pH. How does it resist the change in pH? By making an equilibrium between positive charges and negative charges. So which property of ionic compounds makes them suitable for use in biological buffers? Here they're saying the ability to form covalent bonds with water molecules, no. Their solubility in organic compounds, no. The ability to dissociate into ions that can react with the acids or bases. So when you get an ionic compound, you put it in water, let's say sodium chloride. It is going to separate into chloride ions and sodium ions. So eventually you're going to balance a negative charge and the positive charges, and eventually these would buffer the system, bringing it back to an equilibrium pH. That is one thing that you must be able to understand. So of course the best answer is the ability to dissociate into ions that can react with acids and bases. We go to question 11. They're saying the primary structure of a protein is crucial for the protein's overall function because, so one thing that you must understand whenever you're dealing about proteins, okay, those stages of protein folding, protein structure, and other things. One thing that you must be able to understand is this: whenever you're talking about primary, okay, whenever you're talking about primary, the primary is going to determine how. It determines the how question, how the protein is going to fold. I did not say it determines the protein folding, no. I said how the protein is going to fold. So it's all about preparation. When you talk about secondary, you are simply talking about you are simply talking about the folding of the protein, the actual folding of the protein. Okay? Okay? The actual folding of the protein, and this is where now you bring in the concepts of the alpha helix, which is found in DNA, and beta printed sheets. So whenever you hear beta printed sheets, alpha helix, folding, think of secondary. But how it will fold, think about primary. Tertiary, it is simply three-dimensional structure. So dimensional structure or structure. So make sure you use that concept to answer questions. They like bringing questions on protein folding, proteins, and other things. So take note. They're saying the primary structure of a protein, on question 11, for the protein's overall function, the primary structure of a protein is crucial for the protein's overall function because the primary structure consists of amino acids joined by peptide bonds. Remember, peptide bonds aren't found in primary. Okay? Alpha helix, remember that beta pleated sheets, you're simply talking about protein formation like DNA. Those things are held together by peptide bonds and they are found in the secondary. Okay, so that one is wrong. The sequence of amino acids determines how, yes, see, this B, they're saying the sequence of amino acids determines the how question, how the protein folds into secondary, tertiary, and quaternary structure. So of course, you don't even have to go far because this one answers what primary is all about. DNA determines, no, DNA does not determine because it's not yet formed at that particular time. So of course the best answer is B. So that is how you answer questions in biology. I will go to question 12. Question 12 is saying the formation of alpha helices and beta printed sheets characterize the dash level of protein. So you see, so remember what I mentioned. The primary determines the how, secondary determines folding, and in that folding you're forming what? A protein. Proteins characterized by formation of peptide, of course, held together by peptide bonds, formation of beta-pleated sheets, and alpha helices. So, of course, it is in secondary. You'll find it in secondary. Okay? 3D structure, tertiary. Okay? We go to question 13. Question 13, they're saying the significant difference between fibrous and globular proteins is that. The difference is that one is going to be involved in structural functions, or honesty. The other one is involved in a complex dynamic functions such as the actions of enzymes and also transportation. Okay, that is one difference, a prominent difference that you must be able to know. Okay, so of course the best answer is going to be your B even before you read your questions. Okay, you can master this even if you are not clear. Okay, just in case maybe they repeat one or two, because questions are usually repeated. Which statement best explains how a protein's tertiary structure is stabilized? So whenever you're talking about tertiary structure of a protein, by the way, this is an important MCQ, essay question. They might ask you stages of protein structure formation. Make sure that you know them at heart. It's very important. Here is saying which statement best explains how a protein's tertiary structure is stabilized. It is stabilized only by hydrophobic? No. It is primarily stabilized by covalent bond? No, not covalent bonds. It is only exclusively by hydrogen bonds between amino acids? No. You don't find only hydrogen bonds in proteins. Okay, it is a complex relationship of different bonds. So of course you might find hydrogens, you might find disulfides, you might find hydrophobic interactions. All those things are going to be there. When you start learning about your DNA, you're going to understand. So the best answer is, of course, your D. We go to question 15. Question 15 is saying dash is the chemical reaction responsible for the polymerization of amino acids into proteins. So amino acids, when they react with each other, at some point you discover that they tend to lose this guy here. Okay, they tend to lose water. So this reaction is called a dehydration reaction. Why? Because they tend to lose water, or condensation reaction. You lose water. So this one is very simple. Actually, you just have to go straight. Go to question 16. Question 16 is saying which of the following accurately describes the formation of peptide bonds between amino acids? Peptide bonds between amino acids. Okay, so peptide bonds form spontaneously in aqueous environments without enzymatic help? No. Okay. B, peptide bonds form through the interaction between the amino group of one amino acid, this is very true, and the carboxyl group of another, and that is very, very true. Okay, so I'm sure by now you know how to show formation of a peptide bond, also a glycosidic bond in carbohydrates and other things. Very important that you know how that is going to be formed. So it is a reaction between an amine group, a hydrogen atom of one amine group, and the OH. Okay, the OH. So what I'm simply talking about is this: you're going to have this amine group. Of course, it's supposed to have, let's say, two hydrogen atoms like that, and then you're going to also have this carboxylic acid group. Okay, it's just an illustration. So what I'm trying to say is that the hydrogen attached to nitrogen should react with this hydroxide on the carboxylic group or carboxylic acid group. Then together they are going to form H2O. That is the dehydration reaction we're talking about. And when they do that, what is going to be formed is going to look like this. So you're going to have this nitrogen. Okay, this nitrogen is going to be attached to this hydrogen, and then remember we've lost the hydrogen here, So this hydrogen will be attached directly to a carbonyl group. So it's going to be like this down there. Okay? Because you've lost the hydroxy group. This that we have formed here is called a peptide bond. Okay, so it is formed between one carboxylic acid and one hydrogen, one amine group, or let me just say one hydrogen attached to the amine group. Important to understand. Question 17 saying hemoglobin is an example of proteins. This is not a structural. Hemoglobin is simply for transportation. Okay, because it contains about 4 pockets where oxygen will have to bind, and then it is carried along in the blood, delivered to various cells where gaseous exchange is going to take place, and eventually formation of energy and other things. Okay, so we go to question 18. Question 18, they're saying, which of the following best explains how heat denatures proteins? Oh, by the way, proteins are enzymes in nature. Okay, proteins are enzymes in nature. Okay, so I don't know that concept which says all proteins, let me say all enzymes are proteins, but not all proteins are enzymes. I think I'm correct on that, or it should be the other way around. But what I'm trying to say is most enzymes are proteins, or let me just say all of them. Okay, so whenever you heat a protein, the hydrogen bonds, they gain kinetic energy, and eventually their rate of vibration eventually increases, and as they vibrate they begin to break, and as they break they begin to lose their three-dimensional structure. So the best answer here is simply, let me see, they're saying heat directly breaks the peptide bonds in the protein backbone. C strengthens disulfide, doesn't strengthen anything. Heat causes proteins to gain new secondary structures. No, the best answer is simply A, which says heat increases the kinetic energy of protein molecules, disrupting weak non-covalent interactions. So of course our best answer is simply A. Question 19, they're saying which of the following amino acids contains sulfur atom and is capable of forming disulfide bond? So the one which there's only one, here we've got cysteine, of course, we've got methionine, tyrosine, lysine. There's only one amino acid listed here which contains a sulfur atom. And these questions, you just have to answer them directly, and that is a cysteine. Okay? And that is a cysteine. That's the only amino acid listed here which contains a sulfide bond. Okay? That is very important for you to understand. It's the only one which contains a sulfur atom, actually. That is very important for you to understand. Okay, so it's very important for you to go through in case maybe they repeat one or two. What is the primary effect of protein denaturing on its structure? So of course another question repeated. It causes the primary structure of protein to be disrupted. They're saying that is A. Breaks all peptide bonds in the protein, leading to complete degradation. It affects the protein secondary, tertiary, and quaternary structures, but not the primary structure. So that is the correct answer. The primary structure, remember, it determines the how. Nothing has begun to happen. No bonds have been formed. Nothing is attaching to nothing. In the secondary, you form a alpha helix attached by hydrogen bonds and beta pleated sheets, which may be attached by different. You might even form DNA and other stuffs attached by different bonds. Okay, you find disulfides, you find hydrophobic bonds, you find covalent, weak covalent bonds there. All those things should start after the secondary phase, going upward. Okay, don't include the primary. So of course, the best answer is C, because it will affect, of course, secondary, tertiary, quaternary, but not really the primary. The primary determines the how only. Okay, so at this point, it's not yet formed bonds when talking about primary. So that is one thing that is very, very important for you to understand. Okay, so we meet in the next session where I'll be answering section B of biology, and of course I'm going to add more questions, high yield questions that might be added in your essay questions. If you found this video interesting and helpful, consider subscribing, hitting the like button, and don't forget to share. Thank you so much.